Welcome to Chapter 4. We finally made it to cells. Cytology is the study of cells. Scientists first became aware of cells when microscopes were invented in 1665 by Robert Hooke. It took a little longer, almost 200 years later, before the cell theory was created. Pictured here, we can see a red blood cell on the left, a little platelet in the middle, and a white blood cell on the right. It took so long because cells are incredibly small. A single human hair, shown here as this blue circle, which is 70 to 100 micrometers in diameter, is compared to the size of a red blood cell, which is only around 7 to 8 micrometers, so much smaller. Smaller than what can be seen with the naked eye. Here are some relative sizes of different things that we've learned about so far. Atoms, lipids, cells, things we know like humans, and it compares it on a scale where we increase things 100-fold. On the far left, measured in nanometers, we see an atom. Coming up to 1 nanometer, we have lipids. They form the walls of our cells. Most cells would be somewhere in that 1 to 100 micrometer range. Then we go up to actual millimeters where we start to see things with the naked eye. This one uses eggs as an example and eventually works its way up to a human, which we measure in meters. In order to see cells, we have to use microscopes. There are 3 varieties, all which give us very different pictures. On the left, we see a light microscope. Light microscope slides are formed by taking cells or a layer of tissue, taking a very small, thin slice, so thin that light is allowed to go through it, then staining that to bring out the color and looking at it in a microscope. This view, unfortunately, does not give us any idea of the 3D shape, and you can only magnify it a certain amount of times. The innovation that allowed us to see cells at an even greater magnification was an electron microscope. But it was still a flat picture. It wasn't until the scanning electron microscope that we were finally able to see a 3D image of our cells. That picture is shown here on the right. Scanning electron microscope pictures are in black and white, but they're often colorized afterwards. Once scientists were able to use the microscopes to see cells, they realized that there were a whole host of different shaped cells. Looking at this picture, the top left cell is a nerve cell. It's very irregular shaped. It has a long axon and all of these short dendrites. Below that, we have a biconcave disc, a red blood cell. They're not round like spheres. We have cube-shaped cells that line our kidney tubules. We have column-shaped cells that line our intestines. We have spherical cells in our cartilage and cylindrical ones in our muscles. It turned out that all cells did not look alike. Cells are shaped differently for their different functions. Sometimes they create barriers by layering up in number, like your skin. As we go through this PowerPoint, we're going to learn a lot about the different functions of these cells and how their shape is actually a requirement for them to be able to fulfill their function. For example, when you change the shape of those red blood cells, like in sickle cell anemia, they clump and clog up your arteries, and they're nowhere near as good for transporting oxygen. The shape is very important. Let's look at a general example of an animal cell. The major components include a cell membrane. This is that outer wall that protects the contents of the cell. The cytoplasm, which is the liquid contents of the cell. The nucleus, where we store our hereditary information. This is this purple ball in the middle of the cell. And the organelles are all of these, quote, little organs throughout the cell. Just like in your body, your organs each have individual functions. All of these different organelles all have a different function, and we'll learn about them on a later slide. But first, let's start with the cell membrane. The membrane around the cell needs to separate the inner contents of the cell from the liquid surrounding it. It's a lot like a water balloon floating in a pool. There's water inside of the balloon and there's water outside, but we want to keep those separate. Substances like nutrients, oxygen, and proteins do need a way to come into the cell, though. We don't want to block those out. Waste products and secretions that we're producing inside of that cell, we also want to get rid of those. So this special membrane has to keep some things in, let some things out, and basically act as a gatekeeper. The cell membrane is composed of primarily the phospholipid bilayer, that double layer of lipid cells. If you recall, the heads are hydrophilic and the tails are hydrophobic, so they stack up and keep water from going in between. They separate the cytosol, that liquid inside of the cell, and the extracellular fluid, which is the fluid that is outside of the cell. Glycolipids are present in the cell membrane. These are carbohydrate markers. They mark the cell and allow the different ones to identify themselves. Your immune cells will actually travel through the body checking these identity markers and making sure they are actually cells that are supposed to be there. Lastly, we have transport proteins. There are 4 types of proteins that we see embedded in the cell wall. First, we have a transport protein. These are specialized to allow specific substances to travel through, sometimes only in one direction, sometimes they're pumping them across, other times they're actually just letting them flow freely. But it's very controlled normally. Receptor proteins are there to communicate with other cells. Identity markers are the ones that immune system is checking. Enzymes are also embedded in the cell wall. Sometimes a substance will come along and bind to an enzyme and cause a change in it, which affects the interior of the cell, but doesn't allow that substance to actually get in. Comes in handy sometime. Enough with the pictures. Let's see some transport proteins actually in action, transporting things, on the next slide.